Good afternoon and welcome back to the afternoon session of the Canola Innovation Day. We have had a lot more great information to get through. So thank you again for joining here in person as well as virtually in your home and offices. My name is Jitao. I'm a research officer at the National Research Council's Aquatic and Crop Research Development Center in Saskatchewan. I'm your chair for this session. My own background is in plant lipid metabolism and seed quality traits, but I'm looking forward to this session. But before we start, there's a special slight modification of a progress note to add. One of the speakers, unfortunately, could not be here today due to unforeseen issues for traveling. So we'll have two speakers. We will have 15 more minutes allocated to the next session in between. So that's now let's get into the session. The first speaker of this hybrid heterosis and production session is Sakaria Liban of DL Seeds. Sakaria is a research scientist at DL Seeds, a canola breeding company in Winnipeg. Sakaria holds an MSc in plant pathology and population genetics, and is currently pursuing his PhD in genomic selection and plant breeding at the University of Manitoba. So Sakaria will present a topic which is really a good transition bridging to the topics this morning on AI and machine learning. And the topic of his presentation will be hybrid prediction and heterosis with machine learning. Welcome, Sakaria.
All right. Thank you, everyone. So hopefully you're not too groggy after lunch. A bit of a big topic to talk about. So I want to follow up on some of what we heard earlier today and talk about how we can use some of that technology we talked about in terms of machine learning and AI, and especially environmental data, to move forward what's possible in terms of hybrid breeding in canola. To start off, canola is a relatively young species formed from the hybridization of Brassica rapa and Brassica oleracea about 7,500 years ago. But it wasn't really until the modern age in the 1970s that the modern crop was developed by Canadian researchers Dr. Downey and Stephenson with improved seed quality characteristics. Canola today is really a trade term used for a specific quality profile, meaning we have uric acid less than 2%, glucosinolate content less than 30 micromoles, and saturated fat less than 7%. So the success of breeding canola, like most major crops, is really focused on and has been primarily driven by foundational phenotypic selection. And phenotypic selection is not something we're going to get away from even with AI models. They continuously need to be updated with accurate and relevant phenotypic data every field season. And that has really driven the success from the first double low varieties to the high-yielding, high-value crop that has brought us all together here today. So very early on, phenotyping, having been done by farmers and early breeders, have noticed that when you cross parents from diverse genetic backgrounds, you see something called heterosis, or the benefit of diverse parents being crossed together, seeing something unique and more productive than the single parent alone. And it started off with those first cross-pollinating crops such as corn and bananas, and it took longer for hybridity and hybrid systems to be commercially available to self-pollinated crops such as canola. And that was really driven by sterility systems such as Ogura, where you could have a sterile female plant and a male plant that restores fertility to the F1 hybrid. And the improvement we see in hybrids is attributed to something in breeders we call heterosis. This is an example of one female with 24 male combinations. The interaction of genetic loci gives rise to two types of heterosis. And the first is something we call general combining ability, which measures the average effect of that female across all the different parents. And that's generally attributed to additive genetic effects. And the second is specific combining ability, which is specific combinations, how they perform, and that's generally non-additive genetic effects. But it's important to consider both GCA and SCA. In a matrix of diverse crosses as shown here, you can find parents that are a good general combiner, but they don't really produce any outstanding specific combinations. And you can find parents which are poor general combiners, but they can produce that excellent specific combination. And of course, the one we all want, good general combiners that also produce high, excellent combinations. But breeders are really interested in those really high heterosis combinations. And what the question really becomes, how can we understand and how can we unravel the genetic relationships that produce those high heterotic combinations? And it was relatively recently that we could start to unpack these genetic effects. The discovery of DNA in 1953 and sequencing of the first human genome in 2003 and the first canola genome in 2014, less than 10 years ago, are all relatively recent events. But they drove breeding forward by allowing us to identify differences in DNA and start to correlate those differences in DNA, first with specific positions, to look at simply inherited traits. And we could do that in the lab through genotyping technologies and select for qualitative traits such as blackleg or clubroot and even herbicide tolerance. And it wasn't until we started expanding on the scope and genotyping millions of SNPs or the diversity of the individuals across the whole genome or by GBS by sequencing, we could look at DNA variations across the whole genome. And this allowed us to separate out our populations into genetic pools or different genetic populations. And parents from those different genetic pools could then be crossed together more reliably to achieve hybrid heterosis and improve the average general combining ability and success rates of hybrids. And later on, we decided to combine these phenotypic data with genotypic data to try to understand and associate different markers in different positions in the genome with the phenotypic response that we're seeing in the parents. And this allowed us to start measuring those different genomic effects that are underlying that heterosis that we're seeing and try to predict some of these GCA and SCA combinations, even for complex traits such as yield and maturity. And this has led to genomic selection models gaining traction. And part of the reason for that is because hybrid breeding programs with different genetic pools are really suitable to genomic selection. If you have a small hybrid breeding program with, say, 100 parents in pool A and 100 parents in pool B, that's still 10,000 potential hybrid combinations, and that's a challenge to phenotype all of them. But if you have a larger breeding population with, say, 1,000 parents in pool A and 1,000 parents in pool B, now you have the problem of 1 million potential hybrid combinations. And in that case, this is where you would turn something like genomic selection to try to unravel and try to pick the best ones by learning from those genetic associations that you made by combining your genotype and phenotype data together. So when we're making those millions or thousands or millions of predictions of hybrid combinations, we have to look at associations of those genetic markers across the genome and across traits. So you see on the left, you can stack different traits and try to figure out the different positions that are important to each of the different traits and try to predict them in a more holistic manner and try to figure out also the interaction between genes and traits, and that also needs to be taken into account. And these combinations also need to be narrowed down depending on the number of dimensions we're looking at. On the right-hand side there, you can look at a simple three-dimensional if you're only selecting for yield, maturity, and oil. But really, the number of dimensions is the number of traits that you're selecting for. And this continues to grow as market demands grow and more is expected of breeders, not just yield, maturity, oil, and protein, but pod chattering and blackleg and clubroot and maybe pest resistance. And so the scale and complexity of the search space continually increases. Another aspect of genomic selection is how can we deal with the environmental aspect. Environment has always been very important to breeders, but typically it has been for selecting, testing varieties at multiple locations to get an average effect of the environmental effect for a single phenotype. And this works well if your environments are similar enough between years, but can lead to disappointment if your environments change significantly. So we believe a better approach is to incorporate environmental data directly into the model alongside genotype data. And this enables a more complex analysis of the interplay between phenotype, genotype, and environment. So in the study I want to share with you today, we genotyped about 1,500 unique parental genotypes using the Illumina 19K array, and it itself is a subset of the well-known 60K array. And we genotyped 5,000 hybrid genotypes, and we generated those F1 hybrid genotypes by capturing the heterozygous and homozygous interactions. And we used a 5-point scale rather than the standard 3-point scale, and this allows us to capture heterozygous by homozygous combinations as well. And we also included encoded areas with deletions as unique haplotypes rather than having them as null data in the models. And the phenotypic data set is from multi-site, multi-year trials provided by DLC, testing between 2016 and 2020. We have approximately 5,000 data points for those Canadian registration system required traits, such as oil, protein, and glucosinolate content, and a similar 5,000 for those primary market traits, such as yield and maturity in this data set. And for the secondary traits, we range from about 444 hybrids for lodging to about 2,000 hybrids for days to flower. Okay. Turning to our environmental data, in this data set we have 108 site years, each with specific GPS coordinates. And the geographic distribution of our sites ranges from southern Manitoba to northern Alberta, covering a large distance of over 2,000 kilometers, and this naturally leads to large variations in the environmental data that we collected. To give you an idea of the phenotypic variance of these sites, we observed a large yield variance, and this is what Julian was alluding to earlier. And this ranges about 3,000 kilograms per hectare between sites within the same year. So here is each of the 5 years, and 2020 was the most variable year, ranging from as low as 1,300 kilograms per hectare to as high as 5,000 kilograms per hectare. And with our large geographic distribution, what we noticed is that the differences between site means within a single year exceeded differences between the mean of the different years themselves. So that was a big point. And so we collected the environmental data, and we took many different types of environmental data, such as temperature, water, air pressure, humidity, wind. And each line here in these graphs represents a different site year. And we also collected data in the winter to capture snowfall and to capture those starting moisture conditions, because it is an important factor. And what we decided to do is enclose our environmental data and anchor it relative to seeding date. And we created both cumulative windows and sequential growth windows. And in total, we created 792 environmental parameters, which covers the different growth windows and also the different environmental parameters. And to analyze our combined genotypic and phenotypic data, we turned to popular mathematical models that were discussed earlier today. So a lot of these linear models work really well, including genomic BUP and Bayesian analysis variants. And then we compared them to if we can get similar or better performance with these emerging AI neural network models. Running our linear models is really straightforward and required applying these mathematical functions with our input and phenotypic data, and running them in a CPU in known languages such as R or Python, and analysis. And after analysis, you can see you achieve a consistent result with these linear models. However, when you run non-linear machine learning models, it's quite different. You give examples of the desired output along with the input data, and the neural network has to learn to associate those two data together and approximate a mathematical function, as is being done here in this simple example here below. In theory, neural networks, they should be able to approximate any mathematical function given sufficient data and compute resources. And this is why neural networks are known as universal function approximators. And if you have a simple function, there's no need for a neural network since you know exactly the function, you can write it out. But for complex systems, it may be easier to have a neural network approximate that. However, setting up a functional neural network is not easy. It does require a really good data set and compute resources and a functional architecture. When I initially ran neural network models on these, I was getting poor performance, on average a little bit worse than the standard linear models. And then I decided to try to work on optimizing and making these more efficient and trying to make it really look at the underlying structure or the architecture, which is really key. So what I decided to do is I looked at 14 different hyperparameters, and I worked on optimizing these different hyperparameters. And so when you have 14 different hyperparameters, you start testing across a wide range of different combinations, and the search space is near infinite. And so you have to use different algorithms to try to optimize and select the best combination of these 14 different hyperparameters. And this takes a tremendous amount of time to optimize these models so you can get the best performance out of them. In our case, it took about 1,000 compute hours, and it did require GPUs with high levels of VRAM to input all of our large data sets, about 24 gigabyte or 8 gigabyte GPU models. And after this optimization step, what we found is that these neural networks have different optimal values for each trait by parameter combination. Additionally, the relative importance of these hyperparameters also varied by trait. So you can see in those lines, some of them are very important, and some of them have less and less importance for each trait and hyperparameter combination. So when we look at it in terms of a heat map, we can look at which hyperparameters are most important for which traits. And overall, it was the learning rate that was most important across most of the traits. Other highlights include yield and maturity were really impacted by dropout rate. And that's really important to prevent overfitting. The number of training cycles or epochs was also most important for parameters of saturates and days to flower. And interestingly, regularizers were important in protein and pod chatter, and they're useful for limiting the relative effect of any single SNP on the model weights. In terms of the effect of data quantity on traits, we can see that all of our traits, they scaled positively with increasing amounts of data that we put into the model. So we did this by subsampling our data set and running it with smaller and smaller subsets to see how well we could improve. And what we saw is that those low hybrid numbers were getting low Pearson correlation values, similar to a lot of public papers that have come out earlier. But as we scaled up, we start seeing an improvement. But this improvement is logarithmic, so you will get gains, but you need to put in significantly more data as you grow to keep seeing those gains in terms of model accuracy. And so we're being limited by data quantity at this point, and it's also now very important to improve the data quality as well. And it's maybe some of that, what we heard about phenotypic data with AI and other tools can help to make it more accurate in terms of data that goes into the models. And when we compare hybrid numbers between these different models, we also see a similar logarithmic curve. There's less variance between models than we saw the variance that we saw between traits. But we did observe that neural networks, they were on average worse performing at those lower hybrid numbers. And that really does mean you need a critical number for them to actually start outperforming those linear models. Okay. And when comparing the different traits, the traits with the least amount of data, at 444 hybrids, which was lodging, had the least accurate prediction at 53% Pearson correlation, which was to be expected. But the trait with the most amount of data, yield, also had the second most difficult trait to predict. And this underlies that the importance of trait complexity as well, not just the number of hybrids that goes into these models. On the other end of the spectrum, the trait that we had the most success with was protein. And this is due to a combination of good heritability for the trait and a large amount of data that went into that specific trait. And when we look at variance among all the models, we see a 1% difference overall between the worst performing model and the best performing model. So the worst in our case was Bayesian ridge regression. The best was a convoluted neural network, one that was optimized. And among the traits, the trait with the highest correlation protein also had the least variance among the different models. On the other end of the spectrum, oil and yield were most impacted by model choice, although they remained relatively low variance between model choice at 3% and 2% differences among models respectively. So let's say you get a 2% improvement with your CNN models after all of that hard work. What kind of effect do you actually see in terms of prediction accuracy? So what we decided to use, plug in our heritability and our selection intensity and do 10% selection intensity and simulate 10 generations of selection for yield. And yes, if you work at it, you can get a small improvement after 10 generations for these neural network models. And I think that eventually with with a lot of funding and advancements in the field of neural networks, there might even be further improvements if we can figure out other ways to optimize these models. So we talked about how neural networks can have small but measurable improvements over traditional functions, but really it's when you add environmental data that you can take another step forward as well. So to do this, we first looked at collecting that environmental data we had talked about earlier to predict the average yield among our 108 site years and using those 792 environmental parameters. And we're able to do so with some level of accuracy, although it's very poor at 52%. And we had a really high standard deviation at 14%. With our genomic model, what we're seeing with the best performing models, we're getting about 60% Pearson correlation for yield. And this is the combination of those 5,000 hybrids and 19,000 SNPs. So it really is a 97 million cell matrix data set that's going into that. And the standard deviation was reduced to 2.4% as well. But when you combine all three together and we get our full model with environmental data added, we can see that now we're looking at 5,000 hybrids. We're splitting that into 33,000 site year by hybrid combinations. And we're adding 792 environmental parameters on top of our 19,000 SNPs. And so the cell matrix grows to 655 million cells. And this does increase compute resources significantly, but we're also able to obtain a really good improvement, 25% improvement in yield performance using Pearson correlations. And we also saw a similar reduction in standard deviation between the different train test splits we had. And yes, across the different traits, we also saw an average of a 10% improvement, and we moved from 0.53 to 0.79 with, from genotype data alone to the GBAHI environmental data incorporated as well. And what I wanted to know really was what was happening and how much the environmental data was impacting and what in the environmental model was contributing to these different effects. So we can make a little heat map to try to figure that out. And what we observe is that water is the most important trait overall and really contributes to the improvement in the models that we're seeing. We also see some expected results, such as wind being really important for pod chatter, specifically peaks at 100 days post-seeding. But we also saw some unexpected results. We saw that cloud cover was affecting saturate levels and peak at 70 days, and that cumulative air pressure was contributing to glucosinolate contents during the first 6 weeks. So some of this is not intuitive, doesn't make sense, and probably something that plant physiologists can look at further. So to conclude off, I want to say that there is a lot of opportunity ahead in the field of genomic selection. One of the things that's happening is that continuous price drop in terms of sequencing costs. There was a tweet by Illumina that $2 per gigabase sequencing. That's raw cost, not the cost you're going to get, but it still will come down in price as we go along. And eventually, genotyping will be a lot cheaper than phenotyping. Phenotyping will become the bottleneck. And similarly, there's a lot of investment in AI, and there's different tools and different versions and different optimizations that can be done. And yes, it's a lot of work to get it to work and even to get it to go better, and you need a lot of data. But I do think that over time, you might get a slight advantage if there's a lot of more bright people working on this and trying to improve this field. And finally, I think that we can move from just having untested, predicting untested hybrids to predicting untested environments as well. And this should allow us to balance our data sets and mitigate selection inaccuracy due to environmental variance across sites and across years. And it can help us hopefully hedge against the worst of climate change by trying to predict for those untested environments. So with our 792 environmental parameters, we can mix and match sites. So we can have a site with the early season where there was like an Alberta 2018 site and the late season was like a Manitoba site. Or we can create even entirely new sites based on climate models, what we think is going to happen in 2035, and hopefully select the best genotypes for those new conditions. And with that, I'd like to acknowledge my colleagues at DLSeeds for providing this valuable data set, Supervisor Rob Duncan, committee members, and collaborators, Julian and Leonard at the University of Gießen, and I'll open it up for some questions.
Thank you so much for speaking on time. We have time for questions, either from floor or online. While we are waiting for questions, and I have one. So you mentioned about the hyperparameters. What is the simplest way to? There is one?
I'll speak into the mic.
Oh, I saw. Yeah. Okay. Yeah. So my question is about what is the simplest way to understand the hyperparameters you mentioned? Is it just a mathematical expression of a multiple factors with?
So neural network models, like most things, need some sort of settings or some sort of methodology in terms of analysis. In most cases, Bayesian and linear regression models are quite simple in terms of how the architecture is designed. But for neural network models, you have these hyperparameters. The simplest one, perhaps people can understand, is how much neurons we have. So you can have a network with 100 neurons or 1,000,000 neurons, right? You can decide how many layers are between neurons. You can set some ways the activation functions, right? Which mathematical activation function to use. And so all of these settings do have an impact on the final neural network. And yeah, if you plug in just a standard neural network off the shelf and try to use it in your data set, sometimes you don't get the best outcomes. So optimizing all the different options you have when configuring a neural network is an important part.
Yeah, we do have an online question for you. It says, hi Sakaria, how did you find the impact of treatment of phenotype data on the results in terms of prediction? And have you tried feeding raw data or some type of means to the models? Thank you.
Yeah, I did try putting in raw data originally. And then afterwards, I used means adjusted with a mixed model, try to average it out. I definitely think you do need some sort of adjustment, and especially when you're trying to build out these larger data sets. You're not going to have all of your hybrids in one location in one site, so you need some way of adjusting to means. You can have the original data that I showed with the 60% was just with the means data. And then, yeah, when you add the environment by means, it's a different model at that point.
Yes, Mark, there's another question from the floor. The microphone here. Thank you.
When you are optimizing models for different traits, are you taking into account those traits that are controlled by major genes, or you consider all the markers as random effect, or you put those loci as fixed effect to get a better prediction?
Yeah, so for these neural network models, you don't actually tell the model which ones are dominant or not. You can have some situations where a single SNP or a single marker will adversely affect the model weights. But that's where some of that tuning helps to try to adjust for that. But yeah, it depends on the trait. Some traits will have one marker being really important and the rest being secondary factors. So it is a problem of just testing out what works best within each model. I don't really set ahead of time. I think these are the best parameters to do. I just have the search space open and figure out which parameters can do well when you have your test train splits.
So if you have more questions, please reach out to Sakaria or through either directly to him or through other contact. If there's other questions from the online, we'll reach him as well. Thank you very much, Sakaria. So our next speaker is Dr. Bifang Cheng. Bifang earned her bachelor's and MSc degrees from the Huazhong Agricultural University, and she pursued her PhD in the Swedish University of Agriculture, where she studied cytogenetics and plant breeding. She has worked for a number of years in bioregional for very long-chain fatty acid biosynthesis production, and she has since joined AFC now on the development and high-yield brown mustard hybrid varieties. Now the topic of her presentation today is enhancing condiment mustard yield by exploiting heterosis. Bifang.
Thanks, Jitao, for the introduction, and thanks to the organizing committee for giving me the opportunity to talk about the condiment mustard in the Canola week. This one? So first, I will give you a short introduction on condiment mustards. Then I will talk about hybrid system establishment and breeding in mustard pigeon seed. Yield enhancement by exploiting partial heterosis in composite variety in yellow mustard. Development of group 2 herbicide immune-tolerant pigeon seed. Molecular characterization of the fatty acid enagase FAE1 gene in yellow mustard. There are 3 types of condiment mustards. Yellow mustard, species name Synapsa alba, is yellow-seeded, and many used to make hot dog mustard. Brown mustard, species name Brassica juncea, and used to make Dijon mustard. Oriental mustard is yellow-seeded Brassica juncea and used to make wasabi in Japan. Condiment mustards are planted on 300,000 to 500,000 acres annually in Western Canada, with a farm gate value of $50 million to $100 million. Canada is the leading exporter of condiment mustard in the world. USA and Europe are the big markets for yellow mustard. Brown and oriental mustards are exported to France and Japan, respectively. AFC Saskatchewan Research and Development Center has been responsible for condiment mustard breeding since the 1970s. Brown and oriental mustard is a self-pollinated crop. Pedigree breeding has been used for cultivar development in this crop. Quality traits such as protein content have been increased, but seed yield remains stagnated. Yellow mustard is a cross-pollinated crop due to its self-incompatibility reproduction system. Recurrent selection has been used for genetic enhancement of this crop. Quality traits such as protein and mucilage contents have been enhanced, but seed yield is not increased via recurrent selection due to its low heritability. In view of the breeding challenges, we have directed our breeding approach and effort towards developing high-yield composite varieties in yellow mustard and hybrid varieties in brown and oriental mustard by exploiting heterosis since I joined the AFC SRDC in 2009. Now I will talk about hybrid system establishment and breeding in mustard pigeon seed. The Ogura CM's hybrid system in radish was transferred into Brassica napus, and then from Brassica napus into Brassica juncea via interspecific hybridization by the researchers at INRA, France. AAFC SRDC obtained the B-Jung-Xiao Uyghur CMS hybrid system from INRA in 2003. The B-Jung-Xiao Uyghur CMS hybrid restore line carries a large radish fragment linked to the restore gene we call the radish linkage drug, and the brassica napus fragment linked to the radish fragment we call the napus linkage drug. As shown in the picture on the right side, the B-Jung-Xiao Uyghur CMS restore line has a poor growth vigor and poor seed setting due to the linkage drug. As a result, the B-Jung-Xiao Uyghur CMS hybrid system from INRA is not functional. Molecular markers specific for the restore gene, and also 10 markers for the radish fragment, and 5 markers for the napus fragment in the restore line have been successfully developed by different researchers. These markers can be used for marker-assisted selection of improved restore line with reduced linkage drug. The bigger challenge for us is how to reduce the linkage drug in the restore line. We have designed 3 approaches to tackle the linkage drug issue in the restore line. Approaches 1 and 2, conventional marker-assisted selection. Approach 3, induced homologous chromosome reaccumulation involving the radish fragment. Approach 1, the restore gene of the B-Jung-Xiao Uyghur CMS restore line was transferred into condiment B-Jung-Xiao background. Over 900 plants at different generations were genotyped using the 10 radish and 5 napus markers. All the restore plants carried the entire radish fragment, suggesting that homologous reaccumulation involved the radish fragment did not occur in the condiment B-Jung-Xiao background, and this approach did not work out. Approach 2, the restore gene of the B-Jung-Xiao Uyghur CMS restore line was transferred into Canola B-Jung-Xiao background. Over 900 plants at different generations were screened using the 10 radish and 5 napus markers. In the BC3 progeny, 1 restore plant was found to have lost 4 radish markers and served to produce homozygous restore line. And the resulting homozygous restore line has some improvement in growth figure and seed setting. Approach 3, induce homologous chromosome reaccumulation involves the radish fragment by transferring the restore gene of the B-Jung-Xiao Uyghur CMS restore line into resynthesized B-Jung-Xiao background. As you know, resynthesized B-Jung-Xiao is unstable at meiosis, with very high frequencies of inter- and intra-genomic homologous chromosome pairing. We anticipated that the frequency of inter-genomic homologous chromosome pairing involving the radish fragment could be increased in the resynthesized B-Jung-Xiao background. And this slide shows the breed scheme to develop the improved restore line. The restore plant was crossed with a resynthesized B-Jung-Xiao line. The F1 restore plant was served to produce F2 seeds. In the F2 generation, 100 plants were genotyped using the 10 radish and 5 napus markers. 1 restore plant was found to have lost 1 napus marker and served to produce F3 seeds. In the F3 generation, over 200 plants were genotyped using the 10 radish and 4 napus markers. 1 restore plant was found to have lost 2 radish markers and served to produce F4 seeds. In the F4 generation, over 100 plants were genotyped using the 8 radish and 4 napus markers, and 1 restore plant was found to have further lost 6 radish markers and 3 napus markers, and served to produce homozygous restore line. As shown in this slide, The improved restore line containing only one neighbors marker and two registry markers and the restore gene has a strong growth vigor and good seed setting, which is comparable with the Czech variety on the left side, Catanus. The development of the improved restore line has made the Agri-Food hybrid system fully functional in Brassica juncea. Hybrid breeding in brown and oriental mustard was initiated in 2013 after we had developed the improved restore line. The first brown mustard hybrid variety, AAC Brown 18, was registered in 2018. And this table shows the summary of agronomic performance of AAC Brown 18 from 2017 to 2022 over 47 station years data. On average, AAC Brown 18 has a significant high, 19% higher yield than the Czech variety Centennial Brown, the same maturity as Centennial Brown. Now, I will talk about yield enhancement by exploiting partial heterosis in composite variety in yellow mustard. Yield increase is very challenging in yellow mustard. As you know, yellow mustard has only 5 to 6 seeds per pod. It is difficult to fix desirable traits in a breeding line due to self-incompatibility. There is no hybrid system available in this crop. Therefore, we decided to enhance yellow mustard yield by exploiting partial heterosis in composite variety. Composite variety is a mixture of 2 or more parents and naturally formed hybrids between those parents. Approach to developing composite variety. First, diverse partial self-compatible inbreed lines were produced while pedigree breeding of heterozygous plants of deepened open pollinated population varieties of yellow mustard. The agronomic performance of the inbreed lines was evaluated in the field nursery. Inbreed lines with strong growth vigor, good agronomic performance, high yield potential were DNA fingerprinted using genotype by sequencing. Unique inbreed lines with similar quantity profile and agronomic traits, but exhibiting genetic diversity, were selected as component lines to make composite lines. And this slide shows that some of the inbreed lines on the left side have a growth vigor as strong as the Czech variety. These inbreed lines were selected as component lines to make composite lines. Development of a composite variety in yellow mustard was initiated in 2011. The first yellow mustard composite variety, AAC Yellow 80, was registered in 2020. And this table shows the agronomic performance of AAC Yellow 80 from 2019 to 2022, in total 45 station years data. On average, AAC Yellow has a significantly high, 9% higher yield than the Czech variety Andenti, the same maturity as Andenti. Development of group 2 herbicide tolerant B-juncea. 2 EME-tolerant lines, B4004 and B4005, were successfully developed using EM's seed mutagenesis approach. Genetic studies indicated that the EME-tolerant trait of B4004 and B4005 was controlled by different gene locus. Cloning and sequence alignment of the ALS genes from the Czech variety Centennial Brown, B4004 and B4005 revealed that B4004 has the EME-tolerant ALS gene located in the B genome, and B4005 has the EME-tolerant ALS gene located in the A genome. Homozygous EME-tolerant line B4017 was produced by combining the EME-tolerant ALS gene from the A genome of B4005 and the B genome of B4004. Tolerance of B4017 to different group 2 herbicides was tested in the field. As shown in this slide, B4017 is tolerant to the group 2 herbicide Odyssey. So the left, the plot is before spraying. You can see many ways in the plots. 2 weeks after spraying Odyssey, all of the weeds died and the plants were growing. 3 weeks after spraying, the plants were at flowering stage. We are currently working on transferring the immune-tolerant trait into the Bijion cell or Gluaceum's hybrid system to develop brown group 2 herbicide-tolerant brown and orange hybrid varieties for the mustard industry. Yellow mustard is a cross-pollinated crop. Therefore, open pollinated population varieties comprise great genetic variation. For instance, 4 Eurasica variants were identified in yellow mustard. Y517 has a high uric acid content, average 52.9%. Y496 has a medium uric acid content, average 24.4%. Y1130 has a low uric acid content, average only 1.6%. Y514 is a zero uric 9. Genetic studies indicated that the high, medium, low, and zero uric contents were controlled by the 4 FA1 alleles, E1, E2, E3, and E3, respectively, in yellow mustard. To reveal the molecular mechanism controlling the uric acid content, we have cloned and sequenced the 4 FA1 alleles. This figure shows the alignment of the coding DNA sequences and 5 flanking sequences of the 4 FA1 alleles. E1 represents a fully functional allele with a coding DNA sequence of 1521 base pair and a promoter located adjacent to the CDS. E2 and E3 originated from an insertion of 4863 BP retron transposon in the 5-UTR. The increased distance between the promoter and the CDS might lead to the reduction in the transcription level of E2, E3, and thereby decreasing the uric acid content. E3 is an epi allele with CG methylation in the promoter region, suggesting that DNA methylation in the promoter led to the reduction in the transcription level of E3, thereby decreasing the uric acid content. The zero uric allele resulted from insertion disruption in the CDS by 3,100 BP DNA transposon. And this table shows that the CG methylation in the promoter region of the E3 is on the lower part of the figure, and no DNA methylation was detected in the promoter region of E2. The FA1 gene case study revealed that transposable element insertion and epigenetic modification generate multiple alleles, varying in expression level, and modulate the trait phenotype from qualitative to quantitative variation. Development of canola quality yellow mustard with high stability oil. Different breeding lines with zero uric, zero hydroxybenzoglycinate, low linoleic, and low linoleic acid contents have been developed in yellow mustard at AFC SRDC. The availability of these gene sources makes it possible to develop canola quality yellow mustard with high stability oil. Moreover, yellow mustard is inherently drought-tolerant, foliar beetle resistant, Shearer resistant, and resistant to diseases such as black leg, and has a strong early vigor. Development of yellow mustard as an oilseed crop will lead to crop diversification and could provide the canola industry with a resilient and sustainable future. Breeding work is a teamwork. So acknowledgments go to the master breeding group, And where you see the marker and the chemistry lab, and the funding support. And thank you for your attention.
Thank you very much, Bifang. I think we should have time for 1 or 2 questions, please. Yeah.
What's your oil content on yellow mustard?
As you know, yellow mustard is a high protein, low oil content. So average oil probably about 26 to 27%. Protein content is can be 45%. And yellow mustard also used as make a hot dog mustard. And also it has been used as a plant protein in the sausage. So there's a company, because the yellow mustard contains hydroxybenzoglycinate, when they use yellow mustard seed as a plant protein in the sausage, so they have to heat the mustard seed to kill the myosines. So then they can use the mustard seed as a plant protein in the mustard seed. So I think the canola quality seed would be very good as a protein source.
There's a question from the other end on the floor.
Hey, thank you very much, Bifang. Obviously, I was excited by that last slide where you said flea beetles. But what about the canola quality of the mustard? Like you said before, Synapsis alba doesn't produce that many seeds. Are we looking at a big yield difference between canola quality canola and canola quality mustard?
Yeah, I think you need a breeding work right now, and canola, right now we have only the breeding line, is the inbreeding line with the canola quality trees. So I think you need a breeding work to increase the yield. If you really put effort, you can probably increase the yield. Yeah. I like yellow mustard very much because we usually have Junseil trials side by side with yellow mustard trials. So last couple of years, so most of our Junseil trials were damaged by the flea beetles, and then the yellow mustard seems the name of the yellow mustard known the flea beetles. Yeah, yeah.
Thank you, Bifang. Yeah, please join me in thanking both speakers for their excellent presentations. I would like now to call Hossein Borhan to chairing the final session of the day.
Good afternoon. My name is Hossein Borhan. I'm research scientist with Agri-Food and Agri-Food Canada in Saskatchewan. I'll be your chair for the next session, that is gene editing, a subject that has been really seeing quite advances in the past decades and has been probably one of the hottest topics in biology, and also promised to probably provide solution to many challenges that we face in different biological platform. So we have the talk today. We have 3 talks, and they are both dealing with the science aspects of and technology aspect of the CRISPR-Cas gene editing, and also the regulatory issue that is also a hot topic. Our first speaker is Dr. Kevin Rozwadowski. I would like to welcome him to the stage. Kevin is a research scientist at Agriculture and Agri-Food Canada in Saskatchewan. He was raised in a mixed farm in Manitoba, and where he environment that instilled a deep interest in him in biology. He pursued his study with started study with a Bachelor of Science in Agriculture with a specialty in plant science from University of Manitoba. He obtained his master in applied microbiology from University of Saskatchewan. Then he pursued to a PhD study in molecular biology and genetics at University of Guelph, and his thesis was on pollen and pistil interaction and recognition. So Kemi will talk about the gene editing system for canola.
Thank you, Hossein, and thank you to the organizing committee to inviting me to give a talk today. Part of that invitation was to provide an overview of, quote, the intellectual property landscape. So the information I'm going to provide to you today was with a bit of a heavy caveat. It's what I'm not an IP expert, so I'll give you a summary of what I've been able to glean from the literature, but I'm happy to talk about that to the extent that I can. And also I'll talk to you about our work to optimize gene editing systems for canola. And I'll give you some examples of our efforts and our successes there. And also I'll sum up with some of our more recent work to accelerate the editing process in canola. So moving on. So CRISPR, I mean, it's been around for a dozen years now. I mean, it's I see it everywhere in the literature and in the media. But just to summarize what it is for those who don't know about it. So we have our genomic DNA, and we have a gene of interest that we want to edit, and then by some means we need to express in that cell the Cas nuclease, which is represented by the red Pac-Man, and a guide RNA, which these two interact to form a ribonucleoprotein complex that then interacts with the genome and scans the genome for the target site. That target site is determined by Watson-Crick base pairing between the guide RNA and the template DNA. And then there's a double-strand break that's formed at that site. That break is recognized by the cellular DNA repair machinery, and at some incidents is repaired in an error-prone manner, leading to insertion or deletion of one or more base pairs that leads to mutation at that site. And typically those insertions or deletions lead to a frame shift, resulting in a premature stop codon, and results in a non-functional truncated protein to result in your null allele. Okay, so the importance of gene editing for the canola development is that it helps us define gene function. So we can identify all kinds of, or a great number of genes based on genomics studies and DNA sequencing, or by mapping, we identify several candidate genes, which one's responsible for our phenotype. So we can use editing to knock it out and determine its phenotypic effects. The other important aspect that I see is translational research. Editing enables us to move traits or to test traits that have been discovered in other crop species, so corn or soy or rice, that we wouldn't be able to do obviously by normal breeding approaches, and to test those traits directly in canola, or also evaluating traits that could be identified from model species such as Arabidopsis, which I'll show you a couple of examples later. So the other good part is that edits are genetically stable, and the outcome will be that there's no foreign DNA sequences in that genotype. So from a regulatory perspective, it's also acceptable. Okay, so there's two main kind of CRISPR systems that are in dominance in usage. Based on the Cas9 nuclease on the left or the Cas12a nuclease on the right. So Cas12a is also known as CPF1, and then the type enzymes are from the two microorganism species, bacterial species represented there. So they're very large proteins encoded by large genes, which can add difficulties in getting sufficient expression to gain edits in plant cells at a high enough, at an acceptable frequency. The guide RNAs are represented in the shaded pictures. You can tell that they're somewhat structurally different as well, and that the cleavage site resulting from the action of the nucleases happens at different spots with respect to the guide RNA. So the Cas9 typically results in a blunt double-strand break, and which repair leads to indels of typically indels of 1 to a few nucleotides, and the Cas12a has sticky 5' overhangs that typically, when they're repaired, they lead to several base pair deletions. Okay, so the IP landscape. Now, so foundational patents were awarded to the institutions of the principal inventors at the time. So at the Broad Institute, linked to MIT and Harvard, where Feng Zhang is. And so they're awarded a patent for use of Cas9 in eukaryotes in the US, and Cas12a in the US and Europe and other jurisdictions. So the second group is known as colloquially as the CVC group, which is a combination of the Nobel Prize winners Jennifer Doudna and Emmanuel Charpentier. And they have their usage of or IP related to Cas9 and eukaryotes in Europe. They've established what are known as surrogate companies that are responsible for their agricultural applications, so Caribou Biosciences and ERS Genomics for the CVC patents, and Pairwise for the Broad patents. So the major international plant breeding companies have licensees from these organizations for use of Cas9 and Cas12a, and so obviously they can commercialize the traits that they develop with them. From what I read in the literature, the cost was eye-wateringly expensive for them. So the one item I saw was $100 million plus royalties on top of that. And but my understanding is that for smaller companies, smaller entities, that there's a scaled price, so it would be less for smaller companies depending upon the footprint of the crops and the acreage. But in this context, for licensees being expensive, where does that leave public research labs or small companies? We can use the systems for research, but commercialization can be a problem. So if we develop some interesting trait, where do we go with it? So enter the MAD7 enzyme developed by a company called Inscripta. So why is it called MAD7? It was identified from a metagenomic screen of populations in Madagascar. And it has sufficient sequence and structural uniqueness from the typical CAS 12A to enable the US Patent Office to award them a patent. So for several years, the Inscripta offered an R&D license that was basically no cost, which is fantastic. And the other aspect is that they enable, through that no-cost license, you to commercialize traits, commercialize products, as long as they don't, those outcome products don't retain the MAD7 enzyme, which isn't a problem because you would cross that out anyhow. So AAFC has such a license for use of MAD7, but the conditions for obtaining this license has changed since January of this year, and you would have to contact Inscripta directly now to inquire about what's the context of the license that they provide. Okay, so the efficacy of MAD7 in plants was debatable. There's an original paper applying MAD7 in a zebrafish model, which is typically cultured at 26 to 28 degrees. And when they tried MAD7, they got no edits. So that's a bit discouraging. But they did get edits when they provided an extended heat shock at 37. So there's some sense to that. It comes from a microorganism from our gut. So it evolved in a temperature environment that's 37, so that's sensible for its activity to be optimized at that level. Also, there's been 2 papers showing that its activity in rice and wheat, but again, its frequency was not outstanding for edits, and they also showed that in protoplasts, they got greater editing efficiency if they heat shocked at 37 degrees. So in my lab, we've been working to overcome this obstacle, and we've developed what we call an improved MAD7 or iMAD7, and I'll be telling you a bit about our results from that. Okay, so now I'm going to give you a couple of examples of our editing work. First, a Cas9 example. So we've applied that to trying to accomplish what's referred into this PNAS paper by Orta, the smart canopy concept for altering plant architecture to improve absorbance of incident radiation to increase photosynthesis. So please like this slide. It took me a very long time to draw in PowerPoint. But so in my caricature of your typical canola stand, we see that there's as backed up in the literature that I found, there's actually a high degree of incident light that's reflected by the flower layer. So that can impede the amount of photosynthetic capacity by impeding the light reaching the lower leaves. Also, developing siliques can interfere with light. And my collaborator at Saskatoon, Raju Solanya Kanahali, has measured this. So where light interception decreases is decreased for the lower leaves. So what we'd like to do is try and increase light penetration into the crop canopy. So how can we do that? So the idea is if we can make the flowers more adherent to the primary stem and decrease the circumference area, I guess, of the silique layer, that we'd be able to increase light penetration into the lower leaves and therefore lead to a productivity increase. So how can we achieve that? It's something that I've been thinking about for 25 years when I was a postdoc in Raju Datla's lab, where we worked on this gene called brevipedicellus. So in Arabidopsis, you can see on the right in the BP mutant that the siliques are significantly reduced, and or the pedicel is significantly reduced, and the sleek has a downward nature. And his lab worked to illustrate that effect in using a transgenic approach where they overexpressed a repressor of BP function to get a similar effect as shown in Arabidopsis. So we applied editing in canola to achieve that phenotype. And you can see on the right that the flowers are tightly adhered to the stem. And throughout development, that is maintained. So on the lower right, you can see that the pedicels are shorter, and there's a general declination of the sleeks, and that's quantified on the left, that the pedicel is significantly reduced in the mutant background. Interestingly, we also observed that there's a greater number of sleeks on the primary stem. So it's quite significant. We have at least 20 or more sleeks per primary stem. So we don't have the data yet, but I'm hopeful that this might also lead to a yield benefit. Okay, so how do we achieve that? So we identified 4 guide RNAs that bind to a region of identity across all 4, or both the A and the C gene of BP, and then we co-expressed all 4 guide RNAs plus our Cas9 nuclease. And in the lower panel, so what we see is so if we drive the construct by EF1 alpha, we've actually got quite efficient editing. So 20% of our primary transgenics showed the BP phenotype, indicating that all 4 alleles of BP were inactivated, whereas this other promoter didn't work to the same degree. So just to describe the outcome of those lines, so for the 2 alleles at the A3 locus for guide RNA1, so we have a loss of a base or plus a base, and nothing at guide RNA2 or 3, and then plus 1 or plus 2 at guide RNA4. And similarly on the C3 allele plus 1, in this case, both alleles of C3 are identical for the edits. But the take home here is that not all guide RNAs are the same. For guide RNA2, we had no events, and for guide RNA3, only events on the C3 genome. So from a deployment perspective, I highly recommend co-expressing multiple guides. Okay, so that's our Cas9 story. So for our MAD7 stories, we're looking at trying to increase seed set potential by creating multilocular sleeks in Napus, and we're doing that by trying to manipulate the action of the Wuschel-Clavata pathway, as represented in the top left there, which is my cartoon of a shoot apical meristem. And it's the balance between the function of Clavata and Wuschel that leads to differentiation of organs, both vegetative and reproductive organs. Okay, so in the literature in RAPA, there's a natural variant of Clavata that's a null allele that leads to four locules instead of 2, and depending upon which paper you look at, you get an increase in seed per sleek of 40 to 70%. And also, there's a natural variant in the Clavata1 gene of Gensea, where again you get a great increase in seed set. And for a more recent paper where they used editing to knock out Clavata3 to get an increase in seed set in seed per sleek in winter type Napus. Okay, so deploying our IMAD7 system, again we created 4 guide RNAs that are bind to a region of identity across both genes, and then we evaluated different promoters to express MAD7 or the guide RNA array. So we did achieve lines with 4 locules as shown by the arrows, but it hasn't been, this material hasn't been characterized very greatly as our BP stuff. But that visual phenotype allows us to easily score the efficacy of the system. So we compared different promoters, as I was mentioning earlier, driving the expression of the two components. And so again, and these are primary transgenics, or the T0s. So we got 3 of 19 with this construct showing the clavata phenotype, 1 of 19 with this construct showing the clavata phenotype. I had to put my slides in last Thursday, so this material has come on stream. So on Monday, there's actually 1 of 24 with this promoter combination as the clavata phenotype. And Cas9, there's 1 of 16. So the take home from this is that the MAD7 system that we've set up is at least as good, if not better in this context than Cas9. So we've been able to overcome, I think, some of the roadblocks for efficiently using MAD7 for editing in plants. Okay, again, so some of the mutation outcomes, similar to before, two alleles at the A4 or the C4 gene. And what you can see is that, so one allele has -6 base pairs, one is -8, one is -6, and one is -9. But again, not all guide RNAs are equal. So these other ones, we got no editing events. And typically, Cas9 only has insertions or deletions of 1 or a few base pairs, but we actually got larger deletions in this context. Okay, so now I'm close to running out of time, so I'll try and go through this quickly. So so our traditional approach is we make a transformation construct, and we transform canola, and we get our events in our primary transgenic, which is good, but it takes time from the transformation perspective. So how can we speed that up? So we're looking at deploying a system originally developed in the Mahfouz lab and extended in the Dan Voitis lab. So where you have we have a platform line that expresses Cas9, and then you transiently express your guide RNA by using a virus vector. And then if you get the edits in the shoot apical meristem, they will be heritable, because that apical meristem will lead to the reproductive structures. So then you just have to screen your progeny for your editing events. So this way you can do your editing just by changing your guide RNA instead of creating the whole transgenic line again. So it should be much faster. So to achieve that, we're working to develop a virus vector to enable the guide RNA expression. In this slide, what we did was we looked at a handful of viruses, plant viruses that infect crucifers, but don't have severe symptoms, because obviously you don't want to kill your plants with your virus vector. And in this case, we're just like in yesterday's talk, we're talking about siRNA. So virus-induced gene silencing is an siRNA type approach to silence genes. In this case, we're silencing phytoene desaturase, which leads to a bleaching phenotype. So virus 1 leads to bleaching in canola. Virus 2 also leads to bleaching, but not as severe. So we've been focusing on virus 1, which is efficacious in Carinata, Juncea, Camelina, and Arabidopsis. So we're using this system for guide RNA expression in our platform lines, but I don't have data to show you yet. We also use this system to express proteins, so a typical Gus marker reporter gene, as well as we use a transcription factor, PAP1, which turns on the whole phenylpropionate pathway, which leads to accumulation of a reddish pigment. And this is a kind of a live cell marker. So you can see the red foci here, showing its functionality here, as well as in roots, and it also works in camelina. Okay, so how does this work? Again, we have our platform lines, and then the idea here is that we co-express a factor that stimulates a developmental programming to lead to a shoot. So we're using our protein-based expression from our vector to express developmental regulators, plus the guide RNA. So if you induce a shoot formation plus a lesion, then you rescue your shoot and you'll have your editing event directly. So we've evaluated a handful of different developmental regulators. So here's the positive controls and expression of using different developmental regulators. So you can see we're changing leaf development with several transcription factors, and we're also inducing ectopic plantlets with these 2 transcription factors. Okay, in summary, I'm out of time, but just to go briefly, so the gene editing landscape or the IP landscape is complex. Licensing can be costly, but it's essential for commercialization of edited traits. The improvement we made to MAD7 shows that it's an effective editing system in canola, and co-expressing multiple guides is important to get your edits as rapidly as possible. And just to acknowledge just to quickly do some acknowledgements. So Brent and May Hijazi in my lab have been instrumental. They're technicians in my lab that have been instrumental in the success of our program. Mackenzie just joined recently. Sefer Rahman has also just joined recently for I didn't get to talk about our protein engineering work for MAD7, and then our other collaborators. So, okay, thank you.
Thank you, Kevin. We are a little bit over time, but we have time for just one question if there is any question from audience. Is there a question from?
Okay, Kevin, thanks for a great talk. So you may not be able to shed more light on this, but I'll pose it anyways. My understanding, going into sort of our science cluster planning, some of the research that we've been sort of working with our value chain in canola, is that the government of Canada was going to do a broad license arrangement with Doudna Charpentier, and that they were quite friendly to government and university research laboratories, and so that there would be quite a bit of freedom to operate for sort of our public research that we've been working with. And then there would be the usual normal commercial licensing would take over when researchers like yourself would have things in place that they would turn over to seed companies. Is that your understanding?
No.
Maybe I should go through my notes and figure out how the hell.
So I'm not privy to that information about what you mentioned about getting the general license.
Okay. So from your perspective,
you attend meetings with other people than I get access to. So.
Okay. So we should have more discussion, I guess, amongst all of us around whether that could be a strategy that could work.
Right.
And who would be leading that then? Okay.
Yeah, so, but it depends on which platform people are using, right? So the Charpentier, like the CVC group, I mean, so you'd be able to use Cas9, but you wouldn't be able to use Cas12a.
Right.
So Cas12a is by the Broad group.
2. We have to work through 2 then.
Yeah.
Yeah. Okay. Thank you.
You're welcome.
Please join me to thank Kevin for an excellent talk. So our next talk is a recorded presentation. It is by Dr. Heather Shearer from Canadian Food Inspection Agency. Heather is the manager of the Canadian Food Inspection Agency Plant Biosafety Office, or PBO, which oversees the environmental release of plant with novel traits. Heather joined the PBO in 2011 after completing a postdoctoral program with the National Research Council Canada Plant Biotechnology Institute. She obtained her PhD from the University of Guelph and MSc from Plant Molecular Biology from Queen's University. Heather will provide an update on regulation on the use of new genetic tools and regulation around it.
This is Heather Shearer. I'm the manager of the Canadian Food Inspection Agency's Plant Biosafety Office. In this presentation, I'm pleased to have the opportunity to share with you some highlights of a recent guidance update for plant breeders who are using new genetic tools such as gene editing. I work in the seed world, but I'm going to start big here and massively overgeneralize on behalf of the entire government and multiple programs. What is our regulatory trigger? Well, our job is to look at products that are both new and different and determine whether they're safe and suitable for their intended use, whether as foods, as seeds, or as one of the many other product types that are regulated in Canada. As innovators, we know that this is also exactly where your attention is focused, on things that are new and different. And this is where the challenge comes up. Innovators need to know when something new and different is different enough that a government safety assessment is required. But new and different is a difficult thing to define. What is new? How different is different? Gene editing is an exciting tool for innovators. As regulators, while we're also excited for you, I can tell you that editing presents us with some challenges. It can be used to develop something that isn't new or different, just better, but also used to make some really significant changes and improvements to the genetics. Now, if we had a method-based trigger, like you use centrifugation, then it would be simple to say what things were regulated, but then that wouldn't be very targeted. So in this talk, I'm going to tell you more about some updates we've made to our programming to help you as innovators know when something is new and different, particularly if you're using technologies like gene editing. When it comes to the pre-market oversight of novel seed, CFIA assesses safety and issues permissions for release into the environment. The Plant Biosafety Office, where I work, sets overall guidance on what's regulated and makes decisions on authorizations. Our counterparts in Science Branch perform the assessment work and set guidance on assessment information requirements. We have 3 key guidance documents that provide plainer language interpretation of the legal requirements that are set out in Part 5 of the seeds regulations. Today we'll focus on Directive 2009-09, which you can think of as the entry point decision. Is this plant novel or not? This is the guidance that we updated. The other 2 guidance pieces are for getting permission to release your novel plant, so those only apply to novel plants. Now, to set the stage for updating guidance, CFIA undertook a comprehensive review of the scientific, legal, and practical considerations, along with taking a look at the approaches that were being developed by other governments. We've summarized our findings from all of this work in a policy rationale that we've published online. You can see that we aren't big believers in vanity URLs in the government, but I'm hoping if you follow that web address, you should be able to browse your way to the report. Key conclusions that you should be aware of. We determined that gene editing techniques can achieve similar results to conventional breeding and don't have inherent risks that would justify a pre-market safety assessment of every product developed using gene editing. Furthermore, we know that breeding a new variety is a rigorous and intentional process where breeders understand the genetic potential of their crop species, where they plan their breeding and selection strategy, characterize their product from end to end, and really monitor every aspect of their new line at every step along the way. So given this context of plant breeding practices and also our own 30 years of experience of assessing novel products, we don't foresee that outcomes of conventional breeding generally require pre-market assessment, apart from those very few products that have a direct potential to impact the environment. And we also believe that regulation really should be based on the characteristics of the product, not on the process that was used to introduce characteristics. So, fast forwarding with that context in mind, we wrote some draft guidance for consultation, held consultation on the draft guidance, heard from stakeholders. We heard a lot, and this is also available online in a What We Heard report. We redrafted our guidance, checking in with key stakeholders along the way, and released a final updated version of the Guidance Directive 2009-09 on May 3rd, 2023. Many things remained constant. This is because the regulatory principles and underlying regulations have not changed. Plants with novel traits are still defined the same way, where the trait must be both new and have the potential for a significant environmental impact. That's the different part of new and different. Product developers also remain responsible for notifying us of any plants with novel traits. We believe that you know your product best and will be diligent about getting any permissions that may be needed along the path to commercialization. We're of course available if you have questions about how the guidance may apply in your case. So here's what you need to know. What did we update? The headline news here is in bold text. We don't anticipate that breeding and editing techniques will generally result in plants with novel traits. This doesn't eliminate your regulatory obligations to notify us if you do notice some impacts on those 5 environmental safety criteria. But what we're saying up front is that we don't anticipate that this is likely. We're also providing some bright lines around when a plant is a plant with novel traits, regardless of the method of development. This includes any plant with foreign DNA that could not have been introduced through breeding, and also all plants with herbicide tolerance traits, as these are traits with direct links to the environmental safety criteria. However, we're familiar with herbicide tolerance traits, and we're now offering a streamlined 60-day review process. We've already, in fact, authorized our first product using that new approach, a white mustard. The updated guidance is focused on field crops and horticultural crops. So for special use cases that are less familiar to us, we ask that developers consult with us for case-by-case guidance. And last but not least, we heard a great deal about the need for transparency, particularly from the organic sector. In the era prior to gene editing, this sector needed to be aware of transgenic plants, which were all novel and listed in mandatory databases of novel products, as well as being generally detectable through commercial testing methods. With editing, though, there will now be crops developed that aren't novel, so they won't be subject to pre-market assessment and publication in our databases. And at the same time, there may not be good testing methods available. So while transparency for things that we don't regulate is outside of our direct mandate, we did underline in guidance the importance of comprehensive participation in industry-led venues for transparency. And because transparency is highly valued by the Government of Canada as a whole, there were also some additional actions taken to address this feedback. Agri-Food and Agri-Food Canada, or AAFC, has a mandate to support market competitiveness. So to complement the novelty guidance updates and meet the needs identified by the organic sector, AAFC established an industry government technical committee that provided advice to the government on non-regulatory transparency measures. This advice was also published online in a report and provided to the Minister of Agriculture. To oversee the implementation of these enhanced transparency measures, AAFC formed a government industry steering committee on plant breeding innovation transparency. Thankfully no acronym with that one. There's representation on this steering committee from across the seeds and grains value chain, along with AAFC and regulatory departments. This group will be reporting to the Minister of Agriculture on the implementation of the enhanced transparency measures. Finally, I will note that my counterparts who regulate novel foods and novel livestock feeds have also been undertaking a similar update to their novelty guidance documents. The Health Canada guidance was published in May 2022, while the livestock feed guidance is still pending. They have recently concluded a public consultation on that guidance. I know we're all looking forward to having all three of these guidance pieces updated and also to seeing what kinds of new innovations will be brought forward in the coming years, helping Canadian agriculture remain competitive and meet future challenges. So with that, I will thank you for your time today. If you have any questions about what I presented, you'd be very welcome to contact me by email. Thank you.
Our last presentation for today is by Dr. Wei Xiao from University of Saskatchewan. He's going to give a brief talk about applying gene editing for improving brassica resistance to clubroot. It would be an online presentation. So, okay. Yeah. Dr. Janvé, are you, if you are online, could you raise your hand, please? We see you. Okay, so we have, as it seems that we have to be out of here exactly at 3 o'clock, so we are pressed for time. We'll move to the next part, that is a panel discussion. We will have, I would like to invite Kevin to come to the stage, and our other panelists will join us online. It is, if we could, Stacy Singer would be from Agriculture and Agri-Food Canada. Heather, that you just listened to her talk, from Canadian Food Inspection Agency, and also I would like to call upon Dr. Daoquan Cheng, who is developmental biology researcher at the National Research Council of Canada, to join the panel discussion. So if there is any question about either from the basically application side and from the regulatory side for the panel, I would like to audience in the floor on the floor or online to join this discussion.
Well, we could kick off with an online question. I think this one's for Kevin, but probably both of you could answer it. It's concerning the BP phenotype. The question is from Cindy. Would there be an environmental impact with regards to disease or shattering by altering the silique to a more vertical angle?
So that's part of our next steps, is to evaluate if there's any beneficial impact on there there's some possibility that that's the case because of there's differential cell development and number of cells that leads to the BP phenotype. So whether that's also occurs in the abscission zone of the replum, we don't know yet, but we're going to assess that in the upcoming weeks.
Guess there's another question, more from me actually. Would the panel like to comment on using this technology to do more than just silencing genes? What else can you achieve with genome editing?
Yeah, so I mean, there's different technologies that are out there derived from the primary machinery of the CRISPR-Cas system. I didn't get a chance to talk about it, but we've used a process called base editing, which allows kind of in vivo protein engineering. So we've used that to establish herbicide resistance in camelina. So that's kind of a directed mutagenesis approach related. So you can specifically modify certain base pair or your targeted base pairs to encode a different amino acid to lead to new functions in your protein. Another newer technology is called prime editing, which allows you to instill novel several amino acid changes up to 30 or more, and it's also highly specific. So those are kind of two approaches. I think just one more thing that's quite exciting. I think is developing novel promoter alleles. So instead of knocking out the coding region, to use editing to change the promoter structure, leading to novel expression levels that can confer unique phenotypes. So Zach Lippman at Cold Spring Harbor has really been the groundbreaker in that, leading to a diverse new phenotypes in tomato as a result of promoter bashing with CRISPR. So.
Yeah, some other technology also is involved in the gene editing. Also, is pretty good. And for example, currently we are approaching to using that haploid inducer, and this technology can. After we use this technology, we can easily get a double haploid. And first we, and also in China now recently also they have double haploid inducer in Nebraska. And currently in RC we are working on the haploid inducer. So if we have the double haploid inducer, that's to say is we can escape from the tissue culture, that process, or also doubling of the doubling in the plants. Yeah, so such kind of the technology, that's if we apply it in canola, it can be used significantly increase that canola's yard, and also stacked in the traits. Yeah, such like this.
Okay.
I just have a question of, do you survey if there are any off-target editing, and what is the chance of that happening?
So I'm glad you asked that, Jitao, because I don't think off targets are a big consideration in the plant context. off-targets are a primary consideration for medical applications where you want to make sure that there's no secondary mutations. In the plant context, my view is that you're going to back-cross things out anyhow. So if there are off-targets, they're going to be left behind when in your back-cross to when you get rid of your transgene construct. So I don't think it's a real issue. Having said that, it's prudent to ensure that your guide RNAs are gene specific, so one can do genome a blast search to make sure that your guide RNA is specific to your gene of interest. Yeah, but I don't think it's a big issue unless you're looking at a gene family, in which case you want to make sure that you have specificity.
I just have two minor questions. The first one is, what is the plan with susceptibility factors in addressing susceptibility factors in canola? And the other one is, what is the with the plan with identification of genes? Because to do the to be successful with gene editing, we need also to accompany with faster gene isolation mechanisms, either cloning or sequencing. That also in the plan of AFC.
Sorry, I was looking for you in the crowd, so could you, I apologize, could you repeat your questions?
My first question is, what's the plan in addressing susceptibility factors?
So partners for using the system?
Yes, for example, for verticillium, rather than looking only for the resistance gene, if we identify the susceptibility factors and knocking out that gene and make to make it.
Yeah, that's a great idea, right? So that would be certainly doable, is to look at susceptibility genes as well. I mean, I think that's kind of, it's a matter of, if you're looking at doing a large-scale unbiased screen implants using editing, that's not possible. It's used in animal cell culture all the time over the last 5-8 years, looking for synthetic lethals and whatnot. But with some of these new approaches to streamline and multiplex the CRISPR system, you could do kind of a screen for suppressors more practically. I don't think it's practical yet. So, and your second question?
Yeah, just with the gene isolation mechanism, just to edit, you need to first know the genes. So either you do genome association mapping or gene cloning or sequencing.
Right, yeah, so you would need some information, some prior information to design your knockout construct. Yeah. But we can talk more later, for sure.
Okay, so there is one question on the regulatory issue and how that is mainly probably Heather could comment on that. Is that really how this gene editing is going to affect our export market, given that the various countries are the adaptation of the technology is not similar and regulation is not similar in all different countries. So Heather, do you have any insight and comments about this?
First, I'll ask if you can hear me. Yes? Okay. Never sure. So I'll just mention, I don't work in the world of trade. We do work closely with our AFC counterparts who are very much more in tune with the trade environment. But I will say from a regulatory point of view, where the Canadian regulatory approach is shaping up is that the foreign DNA is captured, but the vast majority of edited products are not considered to be subject to these pre-market assessments.
that is in line with a lot of other regulators that are starting to form their process. There's all kinds of different variations on that theme. So sometimes there's notification required depending on what technology you may have used. So every country has their own way to slice and dice that, but Canada really is in with the crowd there. So just as someone who's not in the trade world, I hesitate to speculate on what the trade impacts will be, but from a regulatory point of view, we're very much in step with all of our major trading partners in like-minded countries that are taking that science-based approach to recognizing that editing is a fantastic breeding tool, but it is not something that represents the kind of change necessarily that needs the same degree of assessment as those really blockbuster traits that we would have seen with the GMO technologies. I hope that's a helpful answer.
Yeah, thank you. We had a session this morning about AI, and also it's there's a question about the role of AI in and other high throughput method in trait development and gene editing pipeline. Maybe, I don't know if Stacy would like to comment on that. Sorry.
We're not using AI for anything right now, but I think it has a lot of promise for many things. Phenotyping is one that I think a lot of us can see potential for, that it is starting to be used in that respect for high throughput phenotyping, which I think will make things a lot easier going forward.
So do you think that AI, in terms of basically engineering the Cas proteins and make it more efficient, or basically because delivery of this is also an issue, could AI have a role in that respect as well from technological development aspect of it?
I mean, I think AI could have a place in technology development as well, in terms of potentially screening new Cas variants, that sort of thing. But I don't think we're quite there yet.
Here's a question, I think.
My questions go to Kevin. So it's about the promoter gene editing. So as we know, if you do the gene editing for the coding region, you know which region you target, like a conserved domain, so you know your guidance RNA is designed for that region. So my question is, what is the strategy to gene editing promoter region? So just do randomly, or like a 500 base pair before ATG, or yeah, just want to know this. Thank you.
Yeah, sure. Great question. So again, with reference to the Zach Lippman's work at Cold Spring Harbor, I mean, the bottom line is that in plants is that we don't know all the binding sites for transcription factors. So it's not only the transcription factor binding site, but it's also the spatial interaction between different transcription factors. So in a promoter. So what they did was they used actually I think it's actually they co-expressed a relatively small number of guides targeting the promoter, and then they basically used an unbiased approach for generating promoter alleles that then they evaluated. So in their work in tomato was quite impressive. So they generated a large suite of mutations and evaluated in the field, and they actually looked at manipulating this clavata pathway that I showed in my slide. The good thing about tomato is that it's diploid, so when it segregates out, you can get homozygous promoter alleles. And of course, in NAPES, it's a bit more difficult. So because those promoter alleles would probably be masked by the wild type allele or the promoter of the homeolog. So it's tougher in NAPAS, but I think it'll be an exciting venue in the future.
I mean, we are talking about the acceptance of gene editing, and I think one thing that makes public acceptance more likely is that food shortage. So with all the climate change, and so how do you think that gene editing could basically eliminate the concern regarding climate change and food production? So, would it basically be a enough good reason that for the public to have the acceptance of gene editing approach? So this is to any of the panel members that feel comfortable to answer the question.
I can start this one if you want, Kevin, or you can. It's up to you.
No, I was going to recommend you because I know you have some projects in this area. So.
Yeah, I mean, in terms of eliminating concerns about climate change, I don't know that that's necessarily possible with gene editing alone. I think it's maybe one piece of a puzzle, and an important piece, hopefully. But there are certainly many traits that could be improved using this technology, along with other breeding technologies as well. In my program, we are looking at a lot of traits that are related to getting crops to be better able to withstand climate change effects. So things like drought, salinity, waterlogging, all of these environmental stresses have a massive impact on yields, and in oil seeds, I mean, they impact oil quality, all sorts of different things, seed yields. So it is going to be important, I think, going forward to start focusing on these traits, and hopefully it will help so that we do have enough food going forward.
Okay, so I think we are almost out of time. So I would like to thank all the speakers for engaging in the panel discussion, and also great presentation that they had so far. And also thanks the audio and visual crew at the back that really did a great job. Thank you, all of you, for participating in the session and engaging in the discussion. Thank you. Before we end the session, I think Curtis has some final remarks, so I invite Curtis to the stage.
Okay, to close it up, and we do have a hard stop, so I'll be very brief here. So first of all, thanks everybody for 3 days of great discussion, presentations, etc. We had an excellent 3 days. Day one primarily it typically follows our typical format. Day one being markets in the business climate. Day two being our practical or applied agronomy and research, and then day three being typically our technical breeding day, where we exchange a lot of technical information between genomics, genetics, our public breeders, and our private sector as well. So thanks everybody for hanging in for all 3 days. We had a really large crowd today, and a special thank you also to the farmers who stayed behind for this as well. Proves to be a fairly technical discussion. So thank you very much for all of you for participating in all of this discussion again one last time. Maybe we'll give ourselves a round of applause. All the speakers as well. Again, just to bring things to a close, I'd like to thank again the Canola, the organizing committee. You've seen the organizing committee members up on the screen. A shout out to all of them for all of the hard work. It started along quite a few months ago already, and I think it always proves to be a really good time working on this event and getting everything together. But also a special shout out to Dr. Downey again for attending again, and for all of your energy and and input and guidance again for a great 3 days again. So thanks for being here. And then last but not least, a shout out to all of our sponsors, again for BASF, Bayer, Province Seed, Alberta Canola, Corteva, High Tech Productions, Manitoba Canola Growers, SAS Canola, the Canadian Canola Growers Association, New Seed, Brett Young Seeds, DL Seeds, Syngenta, Western Greens Research Foundation, and Winfield United. So almost a who's who of all of our industry and grower partners in the canola industry. Very much appreciated again for all of the funding support and all of the great input around ideas and bringing this to fruition again. So both ideas and financial support. So thank you very much again. I also just want to give a quick shout out to Sharp, the group that's, Spark. Huh, can't even get that right. Spark has been our partner for the last 2 years. A real big shout out to Spark. They're awesome at doing this, and they make so many things work seamlessly, audio, video, etc. Can't get the name right, my apologies. That won't be a great brand quote, but that's okay. Anyways, thank you very much to Spark again. And there we go. And then one last, a quick shout out to Clint and to Dr. Sally Vale for all of the extra effort that you've put on in bringing this all together. And then finally also to our colleague Taryn Dixon. You've made this your personal project as well to a large extent, and a lot of a lot of hours that go into keeping everything engaged, or everyone engaged, and everyone prepped all the time, and all of your enthusiasm for the entire week. Through all the years, all the months and weeks of planning. So thank you very much for that. Okay. Safe travels home, everybody. I hope everybody makes it home. The weather's not looking great. Thank you. Best of everybody for a safe and wonderful holiday season as well as we go into the holiday season. And see you next year.